It is not at all intimidating to present in person in front of the president of the ASN. For those of you who don't know me, my name is Limonte, and I am an assistant professor and researcher in the Division of Nephrology. And today I will be talking about autophagy and diabetic kidney disease. So I'll begin by describing what autophagy is, first of all, and then I will talk about autophagy and diabetic kidney disease specifically. And lastly, I will share some preliminary data from my K23 on autophagy subtypes in diabetic kidney disease. And what I hope folks come away with is a basic understanding of the importance of autophagy in kidney cellular homeostasis and an appreciation for how molecular data may be useful for identifying mechanistic subtypes of complex heterogeneous diseases like diabetic kidney disease. So without further ado, what is autophagy? So autophagy is a highly evolutionarily conserved catabolic process that occurs inside cells. The purpose of autophagy is to degrade cellular materials, damaged mitochondria and other organelles, protein aggregates, lipids, etc. These in turn can be recycled and reused by cells or eliminated completely. Autophagy is triggered by cellular stress, starvation, reactive oxygen species, inflammation, and overall promotes cellular survival. Damaged mitochondria or protein aggregates sitting inside cells can cause oxidative stress and cellular dysfunction. And autophagy gets rid of these toxic products before they could injure cells further. Autophagy is a dynamic process. It begins with formation, does this work yet? With formation of a membrane called a phagophore, and the phagophore encircles the cytoplasmic cargo, forming an autophagosome. This then fuses with a lysosome, which contains proteases and other enzymes, forming an autolysosome, and it is within that lysosome that degradation occurs. Autophagy is a complex process, and many proteins and signaling pathways are involved in initiation of this process, formation of the autophagosome, and fusion of this with a lysosome. Autophagy is typically studied in laboratory settings as a dynamic process. And so to understand autophagy in static tissues like human kidney biopsy samples, an understanding of the transcription factors and proteins involved in each of the steps in this pathway is necessary. But I won't belabor you with these. This is for my own personal reading and enjoyment. Nutrient deprivation is a major trigger for autophagy. Starvation triggers signaling pathways, activation of sirtuin-1 and AMPK, and inhibition of mTOR that lead to transcription of autophagy genes and mobilization of proteins that are involved in the various steps of autophagy. Other triggers for autophagy include oxidative stress and inflammation. Overall, autophagy is considered a protective process. Cells need just the right amount for ongoing function and in the setting of injury repair. In acute kidney injury, it's well known that autophagy is activated in proximal tubular epithelial cells and other kidney cells and protects against cell death. However, persistent activation of autophagy can actually lead to maladaptive repair and tubulointerstitial fibrosis. Interestingly, autophagy is linked to aging. Autophagy impairment is seen in neurodegenerative disorders and diseases associated with premature aging. We know autophagy activity decreases with age. Upregulation of autophagy promotes longevity. I mentioned that autophagy is triggered by starvation. So this is one of the mechanisms by which caloric restriction and intermittent fasting is actually believed to contribute to longevity. Protein misfolding and aggregation and organelle damage occur with age. So all of these things are ameliorated by autophagy, which is why it's important. Now, what about autophagy in diabetic kidney disease? So plenty of research, mostly done in laboratory models, has shown that autophagy is impaired in diabetic kidney disease. I mentioned autophagy is triggered by starvation. You can think of diabetes as kind of the opposite. It's a state of nutrient excess, hyperglycemia. So instead of starvation triggering activation of sirtuin-1, AMPK, and inhibition of mTOR, in diabetes, we see the opposite. We see inhibition of sirtuin-1 and AMPK and activation of mTOR, which lead to a reduction in autophagy initiation. The hyperglycemic milieu also directly affects autophagy machinery and lysosomal function, causing autophagy impairment. This has been shown in many animal models. And interestingly, this hasn't been studied extensively, but the few studies that have examined how autophagy changes over the course of hyperglycemia have shown that early on short-term hyperglycemia in mouse models and in cells and culture may lead to more autophagy because of cellular stress, but then long-term exposure to hyperglycemia diminishes autophagy ultimately. And now I'll very briefly touch on some of the ways in which autophagy is impaired in diabetic kidney disease. Advanced glycation end products, elements of the hyperglycemic milieu, think of hemoglobin A1C, so hemoglobin with glucose tagged onto it, bind to their respective receptors on cell membranes and trigger intracellular pathways that result in formation of reactive oxygen species and reduced autophagy and lysosomal function. Additionally, intracellular advanced glycation products, which are typically digested through autophagy, are actually harder to digest by lysosomes than non-glycated protein products. And accumulation of these advanced glycation end products and protein aggregates within cells can cause further injury, inflammation, and fibrosis. And you can imagine how this is a vicious cycle, that if advanced glycation end products acting on acting on receptors on the membrane of these cells inhibits autophagy within the cells, and we need autophagy to degrade these toxic products. You can imagine this feed-forward cycle of a lot of intracellular injury. Mitophagy refers to the degradation of damaged mitochondria via autophagy. This is actually one of the main organelles that we see digested in autophagy, and it's because mitochondria are very energetically active and important for cells, especially proximal tubular epithelial cells, that require a lot of ATP generation for active transport. And so if you have damaged mitochondria accumulating within cells, that means more reactive oxygen species, oxidative stress, inflammation, fibrosis, et cetera. So mitophagy refers to the process by which damaged mitochondria are removed via autophagy. There is substantial evidence that mitophagy is specifically impaired in diabetic kidney disease. We know that reduced mitophagy has been observed in laboratory DKD models and also in kidney biopsies from people with DKD. And that is by specifically looking at tissue samples and seeing less of these autophagosomes with mitochondria in them in human kidney biopsy samples. It's been shown that mitophagy inhibitors accelerate senescence, so aging in renal tubular epithelial cells. Hyperglycemia triggers pathways that inhibit mitophagy, and overexpression of mitophagy regulators protect against tubular cell injury. So a lot of evidence that this pathway is important in diabetic kidney disease. And in addition to reduced autophagy induction and impaired activity of autophagy machinery, lysosomal dysfunction is also seen in diabetic kidney disease. As I mentioned, advanced glycation end products may reduce lysosomal biogenesis and impair degradation. Urinary protein overload, albuminuria, and intracellular lipid accumulation may also affect lysosomal pathways. And actually, in kidney biopsies of people with diabetic kidney disease or with diabetes, you can see these lipid vacuoles within proximal tubule cells. Those lipid droplets are digested through lipophagy, is what it's called. And so accumulation of that can cause lysosomal dysfunction through impaired lysosomal membrane formation and acidification. And lastly, reactive oxygen species may induce lysosomal membrane permeabilization. And actually, in one of my first research projects during fellowship, we identified a lysosomal enzyme, cathepsin D, as associated with rapid eGFR decline in type 1 diabetes. This work included a series of orthogonal studies that demonstrated that not only was there increased urinary cathepsin D associated with rapid eGFR decline, but also increased cathepsin D expression in proximal tubular epithelial cells in people with diabetes and chronic kidney disease. And that expression of cathepsin D in proximal tubular epithelial cells was associated with tubular interstitial injury. And so this work was published in Diabetes Care in 2022, and this was actually my first foray into the world of autophagy and the reason that I chose to examine this pathway for my K23. I've talked a lot about autophagy and how it is a conserved important pathway for homeostasis. So what can you do about it? In diabetes models, It's actually been demonstrated that glucose-lowering agents, metformin, SGLT2 inhibitors, GLP-1 receptor agonists, and DPP-4 inhibitors have all been shown to improve mitochondrial function and reduce oxidative stress by enhancing autophagy. And if you think about it just in the most basic way, that autophagy is triggered by starvation, diabetes is nutrient excess. You can see how these glucose-lowering agents create, the illusion of starvation and enhance autophagy. This is one of the mechanisms by which SGLT2 inhibitors have been posited to have renoprotective effects. But interestingly, we see this effect a lot with metformin in laboratory models with activation of the AMK pathways in autophagy, but that hasn't necessarily translated to clinical practice where we see metformin helping with chronic kidney disease. There are also other pharmacological agents that are able to augment autophagy. Actually, the resveratrol, I might be pronouncing that wrong, found in the skin of red grapes, protects against autophagy or induces autophagy. And I think this is one of the reasons why people were saying that wine is good for health. Now I'm going to transition to sharing some preliminary data from my own research examining autophagy and diabetic kidney disease. This work uses data from the Kidney Precision Medicine Project, which we all know is familiarly as the KPMP. And as most of you know, the KPMP is a national research consortium obtaining kidney biopsies in people with common causes of chronic kidney disease, diabetes, and hypertension, as well as acute kidney injury, with the goals of furthering our understanding of disease mechanisms and identifying novel mechanism-based disease subtypes. Kidney biopsy samples undergo comprehensive molecular phenotyping with transcriptomic, proteomic, and metabolomic analyses, generating a wealth of data to be used to understand molecular mechanisms underlying these common forms of kidney disease. Thus far, the KPMP has enrolled and performed kidney biopsies in 66 people with AKI, 242 people with CKD attributed to diabetes or hypertension, and 18 people with diabetes without clinical evidence of kidney disease. This is the DMR group. Those who have been itching to use KPMP data will be excited to hear that as of March of this year, there's a new trove of data openly available on the KPMP website, accessible through this link here. And this includes not only single cell RNA sequencing data that anyone can download, but also biomarker and SomaScan data in blood and urine. And this most recent data release is what I use for the work I'll be showing today. So yes, I generated all of this within the last 2 weeks, so it is very preliminary. This is the first aim of my K23 to define clinical, molecular, and histopathological patterns of autophagy and diabetic kidney disease. And to do this, I've taken 32 adults with CKD and diabetes and 25 controls without CKD or diabetes from the KPMP for whom single-cell RNA sequencing data is available. And specifically, I'm focusing on single-cell RNA sequencing data from kidney proximal tubular cells. And this is because these cells are, as I mentioned, very energetically active, and the first place that I wanted to examine the role of autophagy. And so what I did was I compared expression of autophagy-related genes in proximal tubular epithelial cells between those people with and without diabetic kidney disease. And then I clustered participants with CKD and diabetes by their patterns of autophagy-related gene expression. And what I'm mostly going to focus on are results of that clustering analysis. I showed you that autophagy is this complex dynamic process that has those different stages: initiation of autophagy, formation of the autophagosome, lysosomal function, and fusion with between the lysosome and the autophagosome. I focused on a set of 604 autophagy-related genes that were described in this comprehensive paper by Bordy et al., published in the Nature Family publication Cell, Death, and Disease in 2021. This paper also happens to nicely sort all of the genes involved in autophagy by their specific role. So we have our autophagy regulators, genes involved in mitophagy, docking and fusion refers to the fusion between the autophagosome and the lysosome, et cetera. So these are the baseline characteristics of the DKD and reference KPMP participants with available single cell RNA sequencing data. So for those of you interested in who will ultimately be using the single cell data, this is everyone with diabetes and CKD in it. I want to acknowledge that while the sample size may seem small, 32 people with diabetes and CKD, this is actually pretty substantial for single cell RNA sequencing data. So focusing on this diabetes and CKD group, which I've shortened to DKD for space in my slides, about half of the participants are male, 13% identify as Asian, 19% as Black or African American, 63% as white. Most participants are over 50 years old. In the KPMP data, you'll see age and diabetes duration binned for privacy purposes, and you would need to request the specific numbers separately. There's a range of diabetes durations here, pretty evenly through up to 4 years to greater than 50 years. Most people with diabetes have hypertension, all but one in this cohort. 78% are using RAS blockade. And you have a good distribution of A1Cs, about 1/3 with A1Cs less than 6.5, so you could call this controlled. 1/3 with an A1C of 6.5 to 7.5, and 1/3 with an A1C greater than 7.5. And the mean eGFR in this cohort is 48. We have both eGFR determined using creatinine and cystatin C and creatinine alone, and the median UACR is 500. So this is what our diabetes and CKD cohort looks like. And these biopsies were adjudicated by a team of nephrologists and kidney pathologists who established primary clinical histopathologic diagnoses. And among these 32 people with diabetes and CKD, who I want to reinforce that the KPMP is enrolling people who we would not normally think of biopsying because they only have diabetes and chronic kidney disease, and you assume that that's what's going on. So in this cohort, about 1/3, 34% of people had a primary diagnosis of diabetic nephropathy. 22% had hypertensive nephrosclerosis, and 22% had non-specific or mixed findings that precluded a clear diagnosis of either of these pathologies. 13% had other findings, surprising findings, IgA nephropathy, fibrillary GN, et cetera. But I just think it's really notable that, among this group that we think we know what's going on, it may be more complicated than it appears on the surface. So now looking at our gene expression. So comparing people with diabetes and chronic kidney disease to our controls without diabetes or chronic kidney disease, there was differential expression of 326 of my 604 autophagy-related genes. 252 of these were upregulated, and 74 were downregulated compared to controls. And so you can imagine that, kind of going through each of these and matching these to where they're involved in autophagy is kind of the next step for me. Among people with diabetes and chronic kidney disease, I then compared people with a primary diagnosis of diabetic nephropathy and those with a primary diagnosis of hypertensive nephrosclerosis. And in this group, there was a smaller set of differentially expressed genes, 30 out of the 604. And to my surprise, cathepsin D was actually at the top of the list, distinguishing these two histopathologic diagnoses with greater expression in diabetic nephropathy, which was consistent with the earlier work in my fellowship. What I have here are violin plots of the top 6 differentially expressed genes between these two diagnoses. And 4 out of these are happen to be involved in lysosomal function. Cathepsin D, cathepsin B are both lysosomal enzymes, and this ATP6 is involved in acidifying the lysosome. So on a cursory review, it seems like maybe something that is distinguishing diabetic nephropathy from other pathology is activation of the lysosome. So onto my clustering work. So what I did here was I clustered individuals by expression of autophagy genes within the proximal tubules. So this is only people with diabetes and CKD. And I basically just asked the algorithm to create 3 groups of people with similar autophagy gene expression profiles. And 3 clusters emerged. Cluster 1 contained 17 people, cluster 2 contained 9 people, and cluster 3 contains 6 people. And so again, these clusters are only generated using gene expression. No clinical data goes into sorting these people together. So when I went and looked into the characteristics of the members of these clusters, a couple of interesting things stood out. For one, cluster 3 has no individuals with a primary diagnosis of diabetic nephropathy. And cluster 2 has the highest proportion of diabetic nephropathy. So in my mind, what I was labeling these potential groups as is, okay, maybe no diabetic nephropathy, and then cluster 1 diabetic nephropathy light, because that one has a more kind of mixed picture. So looking at these clusters and the EGFR between them, EGFR doesn't seem to distinguish between the clusters, but cluster 2, which is what I'm calling my DN group, does have a greater range of albuminuria. Am I disappointed? Maybe, maybe not. We know that EGFR is, not a sensitive marker to distinguish between subtypes of disease, and that's kind of the whole point of the KPMP. So I keep going. Looking at diabetes characteristics in these clusters, this is a plot of diabetes durations, and you could see kind of the whole rainbow between them. Just eyeballing, you see a broad range in each cluster. Maybe, cluster 3, which we're calling the non-diabetic nephropathy cluster, has less diabetes duration, up to 20 years, whereas these others, you see people with longer diabetes durations, up to 30 and 50 years. But more interesting to me is that looking at the A1C distribution in these clusters. So cluster 3, what I was calling the non-diabetic nephropathy group, has the least hyperglycemia, with A1Cs mostly in the 6.5 to 7.5 range. But cluster 2, which is the group with the most diabetic nephropathy, has the highest A1Cs. So the biggest proportion of A1C greater than 8.5%. Additionally, cluster 2 members, the diabetic nephropathy group, appear to have, again, just eyeballing, more severe structural tubular interstitial pathology. So we're focusing on the middle group here. We see a greater percent of interstitial fibrosis, tubular atrophy, abnormal luminal morphology, and interstitial infiltration of white blood cells. Cluster 2 members, again, the diabetic nephropathy group, also seem to have more adaptive, maladaptive, and repairing proximal tubular cells than other clusters. These cell states are annotated by the KPMP and have been described in the KPMP's seminal Nature publication, and are believed to reflect injury at the molecular level. So the way I'm kind of putting this together is that my autophagy genes created 3 different subgroups of people based on autophagy that map onto important, clinical phenotypes, diabetic nephropathy versus not, that do seem to match up with hyperglycemia and evidence of cellular injury, even though we're not seeing differences in our traditional biomarkers, EGFR, in these groups. So to try to understand what distinguishes these clusters at the molecular level, I identified differentially expressed genes between the clusters. So this is a dot plot with genes on the x-axis and the 3 clusters on the y-axis. So the darker and bigger the dot, the more expression of that gene. And so you can just kind of, eyeball comparing cluster 3, our non-diabetic nephropathy cluster, to the other clusters. It looks different. You're seeing more genes in this first half expressed compared to the bottom, and less of these other genes. So you could see that there's clear differences between these groups, and then less surprising is that there's less differences between the two groups that had people with diabetic nephropathy in them. Comparing cluster 2, the diabetic nephropathy group, to cluster 3, the non-diabetic nephropathy group, I tried to link the genes that differed to specific steps in autophagy. This is just based on like a literature search of each of these genes and the step of autophagy that they're involved in, and whether they were high or low in cluster 2 versus cluster 3. And so green with an H means that they were higher in the diabetic nephropathy group compared to the non-diabetic nephropathy group, and red with an L means that they were expressed less. So on a cursory look, it appears that in cluster 2, there's greater expression of some genes involved in these first steps of autophagy, T-span1, Jun, and actually this is cystatin C, CST3 is a gene that's involved in autophagy initiation. I know we use it for other reasons, but does that too. So increased expression of genes involved in the initiation, but kind of consistently decreased expression of genes that are involved in the fusion of autophagosomes and lysosomes. So these 4 genes all correspond to proteins that are involved in like kind of the T-snare mechanism, or the literal mechanisms that like fuse these 2 vesicles. But overall, more work is needed to understand how the clusters differ in terms of autophagy activity and how this relates to diabetic nephropathy as a whole. And so my next steps for this work are incorporating pathway analyses using programs such as Ingenuity pathway analysis to understand the specific signaling pathways that are linked to my autophagy genes, and how these are also linked to inflammatory and fibrotic pathways. I'm going to be generating summary scores of autophagy gene expression, validating in other cohorts, and correlating expression of these genes with tubular injury biomarkers. And so to conclude, autophagy is a process by which damaged organelles, protein aggregates, and other cytoplasmic materials are degraded. It's essential for cellular homeostasis. Various aspects of autophagy, induction, autophagoid lysosome formation, lysosomal function, are impaired in diabetes and CKD. Changes in autophagy-related gene expression are apparent in diabetes and CKD. And then the results of the clustering work, proximal tubular autophagy-related gene expression patterns may be able to distinguish between clinically relevant phenotypes of diabetes and CKD that do not otherwise differ by traditional markers of kidney function. And so I'd like to thank my mentors here at UW and my K23 mentors, especially Ian, who's been my mentor since I started in 2018. And with that, I'm happy to take any questions.
Wow, Christine, terrific work. So I had a question about, you mentioned the medications and how they affect mitochondrial function. Do we know the effect on autophagy specifically? And were the patients that were included in your sample on any of those medications?
Yeah, that's a great question. So, so we know that SGLT2 inhibitors and metformin activate, and GLP-1 receptor agonists activate AMPK, which we know triggers autophagy. And that specific pathway, not just knowing that they trigger AMPK, has been studied in animal models. People within the KPM, some people within the KPMP are on SGLT2 inhibitors. I don't have that data right now, but one of the I mentioned that one of my next steps is validating in other cohorts. I'm hoping to use data from a collaborator, Petter Bjornstad, who has a cohort of people, has kidney biopsy samples in people who had been using SGLT2 inhibitors and not been using them, and has single cell data in that cohort, a publication examining mTOR activation, which is also linked to autophagy. So my hope is to look in that cohort at how these genes differ among SGLT2 inhibitor, users and non-users. Laura?
This might be a basic question, but I'm just curious, with the biopsy kind of changes that we're seeing pathologically, would we expect more damage in like the tubular interstitial pattern, or?
Yeah, so the reason, so I've focused on the tubular interstitium because my focus is on autophagy gene expression in proximal tubular epithelial cells. Um, the main adjudicated diagnoses are diabetic nephropathy, hypertensive nephrosclerosis, and both of those are primary like glomerular pathologies. That's how they were determined. I would say the cannot determine group had maybe some like non-specific glomerular scarring and mainly tubular interstitial fibrosis, say that couldn't be pointed to any specific cause. So there was that range of pathology, but and in terms of mapping what I'm seeing to specific pathology, we just have tubular interstitial data available, which is why that's what I shared, but I'm interested in mapping findings to glomerular pathology too.
So great work, Daryl. So it looks like when you're doing no diabetic, it looks like the trend is there in regards to the clusters.
Mm-hmm.
If you, but not that much separation. Do you think if you look at like autophagy, like just separation, second component, since autophagy is related to age,
Mm.
take out age, like you like go now age.
So I'll comment on the second comment first. So another one of the analyses I hope to incorporate is a pseudotime analysis that incorporates data from, so it has like a reference sample without diabetes or CKD, this, older or middle age and older adult with diabetes and CKD, and then an intermediate group of young adults with diabetes without kidney disease. And so I'm collaborating with bioinformaticians at the University of Michigan. And so they've already performed pseudotime analysis of genes involved in like glycolysis and other metabolic pathways using that group, and seen kind of trends that go with not age, but like disease state itself. So that's a way that I'm hoping to address that question. And then in terms of mitophagy, one of the other things I hope to do is, I was focusing on this like large gene set to kind of narrow based on specific functions. And then I could also better like summarize those different functions using scores that reflect those functions too. And I was going to kind of, now that I've kind of spent more time learning about these processes, maybe expand upon, the list given in that paper to include other things I find in the literature. Leah.
Just one more.
Oh, sorry.
Christine, are there any human diseases, renal or non-renal, which
Yeah, so we know about a lot of lysosomal storage diseases, but I haven't seen a lot with autophagy specifically to say. And I think part of that is because autophagy is is essential for life. So if you have defects in that process, you may, have problems earlier on.
Thanks so much. Anyone else with questions? I'm sure Christine will.